Ovulatory activity of female goats adapted to the subtropics is responsive to photoperiod. This presentation will be given by Taylor Smith. This is an outline showing the flow of the presentation. Many breeds of goats exhibit a seasonal pattern in reproductive activity to cope with the seasonal fluctuations in climate and food availability. The main seasonal trait is the ability to restrict breeding at a time of year when the most energetically challenging stages of reproduction coincide with annual peaks in food availability. In temperate areas, the environmental factor that provides cueing for the control of seasonal variations in reproduction is well established to be photoperiod. In contrast, in subtropical and tropical areas, the predictive cue is not well identified, and the respective influences of photoperiod, food availability, and temperature probably vary among locations and species. The purpose of this study was to determine if photoperiod is the environmental factor that provides the cueing for the control of reproductive seasonality of local, well-fed female goats from subtropical Mexico. It was hypothesized that the photoperiod is the environmental factor that times annual sexual activity. The objective of the study was to expose female goats adapted to subtropical Mexico to variations in photoperiod encountered at this latitude by subjecting constantly fed animals to alternations between long and short days, changing every 3 months. The experiment was performed on Mexican local goats from the Laguna region in the state of Coahuila, Mexico during the 2 consecutive years. 28 goats that haven't given birth were used, and the animals were allocated to 3 groups balanced for age and body weight. All goats were fed daily ad libitum, and all animals had constant access to water and mineral blocks. The control group contained 12 goats that remained in an open shed under natural day length and ambient temperature, and the experimental groups, each containing 8 goats, were placed in 2 adjacent light-proof rooms under ambient temperature as well. The experimental groups were exposed for 2 consecutive years to alterations of 3 months of long days and 3 months of short days, and photoperiod was regulated by an electric clock. Ambient temperatures in the open pen and light-proof rooms were recorded once a week. Ovulations were inferred from plasma concentrations of progesterone determined in blood samples obtained twice weekly throughout the experiment. Females with progesterone concentrations greater than or equal to 1 nanogram per milliliter were considered to have initiated the seasonal period over which ovulations occurred, and females with progesterone concentrations less than 1 nanogram per milliliter were considered to be in anovulation. This figure shows the changes in period over which ovulations occurred in the control group, shown in the top graph, group 1 in the middle, and group 2 shown on the bottom graph. Gray areas indicated the months when animals were exposed to short days, and the mean temperature in both the open shed and in the light-proof rooms is indicated by the thin line, while the thick line indicates the natural photoperiod changes. I blew up the graph on the top for visual effect, but it shows that the control group displayed large seasonal variations in period over which ovulations occurred, and only had 2 of these said periods. In contrast, these seasonal changes in period over which ovulations occurred were markedly modified in both experimental groups, and they were related to photoperiodic changes with an inhibition and a stimulation of ovulation following exposure to long days and short days respectively. During the study, experimental goats showed 3 complete periods over which ovulations occurred. It's important to note that both the responses to long and short days occurred irrespective of the time of the year, and therefore irrespective of temperature. However, the second period over which ovulations occurred in group 2, which occurred during the hottest period of the year, ceased earlier than those occurring at other times of the year. This data suggests that temperature can modulate the timing of the response to photoperiod. More specifically, exposure to the highest temperature does not prevent occurrence of ovulation, but hastens its cessation in long days. However, this role of temperature is clearly secondary to that of photoperiod, as changes in temperature do not prevent occurrence of the responses to photoperiod. In both experimental groups, the effect of photoperiod on duration of period over which ovulations occurred was consistent throughout the study. Moreover, all females from these groups had ovulations when stimulated by short days, just as control females did during their natural breeding season. The result of the study allow us to conclude that although temperature is a modulating factor of the seasonal sexual activity, Photoperiod is the main environmental cue determining the timing of breeding and constantly fed female goats adapted to subtropical Mexico. This is the article citation, and thank you for listening.